Okay, so we're going to start here under the assumption that you went ahead and watched those short little videos that I linked in the description for video number 2. If you haven't watched those, please go back and watch them and then come back. I will be waiting here. I promise I'm not going anywhere. But the reason I emphasize that so much is that it's really, really hard to explain this stuff without seeing it in motion. Okay, and I am just not technologically savvy enough to make it happen on my own. So instead, I point you to a couple of very, very excellent, well done, professionally done videos explaining what's happening in that loop of Henle. Okay, and I need you to have an understanding of that before we can come back and spend a little more time nitpicking some of those details out. Okay, so you have seen those. Let's go ahead. So what we saw there before, right, was an example of what we call this countercurrent mechanism, right, where we have flow coming down the descending limb and then flow coming back up the ascending limb. And then as we pass from the loop of Henle through the DCT into that collecting duct, we're then again going down one more time in the opposite direction as we head back down past that ascending limb coming down. Okay, and the reason this becomes so important is that we can get what's called a countercurrent multiplier built out of that. Basically, what we're saying here is that because of the reliance on diffusion or movement into and out of the fluids within this duct at this point, the greater the gradient is, the more that will flow, right? And by having it set up so that the highest concentration in the descending limb and the lowest concentration in the ascending limb are set up at opposite ends, we end up having continuous movement in both directions. Like I said, it's one of those things that really works best if you can see it in motion. So please, again, that's where those videos are coming into play for us. Ultimately, then, we can describe this loop of Henle, right, as the countercurrent exchanger, and in particular, the relationship of the loop of Henle to the vasa recta. Now, we haven't really talked about these guys yet, and that's what this slide here is all about. So we've seen here before, right, blood flow comes in, renal artery branches off, we end up with all these glomeruli, and we have flow of filtrate through the tubule and back down, right? But I can pull a different color here, so work. Really should have probably used red for the blood, but I didn't. Blood continues to flow from this glomerulus, right? Yep. Come on, pen. From this glomerulus down, and it's red again. I guide the technology, right? But it continues to flow, and we have a blood vessel that runs in parallel to the convoluted tubule. And that's what this vasa recta is. Okay, it's blood vessels that run parallel to this tubule so that we can again continue to reabsorb and secrete all the way around. All right. Now again, saw before, the descending limb and the ascending limb of that loop of Henle have different properties, primarily with regard to whether water can come into or out of it. In this descending limb, right, the part that's coming down, right, coming down, we're going to allow water to leave the filtrate. Right? And that water, where does that water go? It ends up out here, right, in the medulla, in the pyramid. Okay, so that because water is leaving the filtrate, right, it's coming out of it. The farther down the descending limb we go, the more concentrated this filtrate becomes. On the ascending limb, what we're going to do is we're going to close that pathway up to water. So water won't continue to come in. Right? Whatever water has left, has left. But as that filtrate comes back up towards the cortex, we're going to continue to pump out, in this case particularly sodium. So sodium is now leaving the filtrate. So we got rid of the water on the way down, and now we're getting rid of the sodium on the way up. And we're actually making this filtrate less concentrated. Now, this doesn't make a whole lot of sense, right? Unless you've seen the rest of this part. We're deconcentrating, I guess. We're making it more dilute, okay? But in return, what's happening here in the ascending limb, the reason that we're pumping out all the sodium is that we're making the surrounding tissues this is where writing in cursive would really be a helpful skill to have remembered from elementary school. Surrounding tissues, get salty, right? So the filtrate doesn't get salty, but the surrounding tissues do. And the reason that becomes important is the final step, right? The very, very final step in this whole process, right? And remember, we passed the distal convoluted tubule, but then we head back down, right, through this collecting duct. And I realize that looks terrible, but we need to move on, right? And the fluid passes down, right? And as it passes down, all of that salt is pulling water out, right? So because we made this tissue salty, it pulls water out of the collecting duct and it concentrates the urine. That's the final step, right? And again, if you saw the videos, that part becomes clear there. The reason we call this the countercurrent multiplier, right, is again, from what we saw in that video, the longer this tube, the greater the difference between the two becomes when it's multiplying it along the length of the loop. Really all it comes down to. Now the countercurrent exchanger, right, the vasa recta, this is where we return water. So if we go back to my little cartoon here, right, I'm just going to pull up a blank screen if I can. Maybe I can, I don't know. There we go. So we have our distal convoluted tubule, right, and our collecting duct. And we've pumped, or we don't pump, it just kind of drains out, it diffuses out water. Come on, there we go. Water is leaving the filtrate, right? This is only half of the process. The vasa recta, right? The reason this vasa recta exists is so that as the ascending limb of the vasa recta, right, blood flows back upward, this whole way around, that water from this tissue ends up back in the blood, right? So the purpose of this vasa recta here is to reclaim that water. Come on, there we go. Vasa recta reclaims H2O from collecting duct, right? And that's really it. So, like I said, there's a lot of complicated things that are going on here. We're trying to keep it as simple as we possibly can, but that's really the gist of the whole thing there. Okay, so at this point then, once we've gone through here, we have had our countercurrent multiplier, we dumped out water first, then we dumped out salt, then we reabsorbed water from the collecting duct into the vasa recta. Okay, what's left then here in the tubule at the end of the day is urine, right? We are now no longer filtrate, we are now officially urine. And urine, I don't think will surprise anybody here what it is. You've all seen urine. It is clearish, at least it's supposed to be clearish. It doesn't mean it's like white, it's like water, but it is clear. You should be able to see through it. If it is cloudy, you might have a UTI. That's one of the initial symptoms of a UTI. In terms of the color, like I said, it's not water clear. Usually it's a pale yellow. The longer it sits in your bladder, the darker yellow it gets. And that yellow color is from a pigment called urochrome. And urochrome is, again, one of those pigments we get from the breakdown of hemoglobin. The longer it sits in your bladder, okay, or the longer it sits in a puddle in the street or whatever, I guess, I don't know, the more of that yellow color we're going to get, right, as it becomes more and more concentrated. Now, there are other symptomatic tools here we can use. One of the reasons we use urine as such an immediate diagnostic test in so many cases is that urine is actually pretty diagnostic for a lot of things. Aside from UTIs, we can look at color. So pink or brown colors usually indicate blood. Smoky colors here usually indicate other adulterating substances. And these are usually from things like the abnormal breakdown of bile pigment, from blood, from things that are dumped in there, either things that you've been prescribed or things that you've not been prescribed, stuff like that. So again, color can be a pretty diagnostic thing. When it comes out the tap, it does smell a little bit. And it's kind of one of those things, the longer it sits, the more it smells, okay? And one of the reasons that it's smelling is that the product that's in your urine from the breakdown of amino acids is something called urea. And urea is really just a stable form of ammonia. Ammonia is something that we can't store in the body because it's poisonous, right? You might remember the whole thing going on now, reminding people not to drink bleach, stuff like that, right? Bleach is ammonia, right? It will kill you. So in order to keep urine in our body so that we can micturate kind of when we need to rather than all the time, we have to convert it to a safe form, and that is urea. Now, as it sits out in the open, bacteria will colonize your urine pool. It is not sterile, but not particularly bacteria rich when it leaves your bladder. But as bacteria colonize the urine, they will convert that urea to ammonia. And that's where the smell comes from. Okay. Depending on what you've eaten recently, there might be other changes to the smell there, particularly, the old joke about eating asparagus. It's true, right? It does make your urine smell slightly different. As far as the pH goes, it is going to be slightly acidic, a pH of about 6, okay? But that can vary downward, can be extremely acidic. 4.5, you would notice this when you feed it. That's really quite acidic. Up to something that's a little more alkaline, maybe something like just in the alkaline side, about 8.0. Again, depending on diet or other things. So the more protein and wheat you eat, okay, the lower the pH is going to be. Right. On the other hand, if you eat primarily a vegan or vegetarian diet, or if you've been vomiting for a really long time, or if you have a UTI, all of those things can cause your pH to go up. Specific gravity is just density relative to water. Okay, so a number above 1 means that it's more dense than water. So your urine is just a little bit more dense than water, pure water, because there's stuff in it. Now again, we've concentrated it. Right? It's not pure water. We've done it as much as we possibly can to concentrate it, but it is still mostly water. It's about 95% water. That number goes up and down a little bit, again, depending on what you've been eating and what you've been taking and stuff like that, but it is still mostly water. It contains our old friend urea, right? We talked about in the last slide. Uric acid is another form of an ammoniac waste product. In this case, we create uric acid from the breakdown of nucleic acids. It's also going to contain creatinine, right, which is the waste product of creatine phosphate. Other stuff that's going to be in there, some potassium, there's going to be some phosphates and sulfates, calcium, magnesium, bicarbonate, all that kind of stuff in there. Any abnormally high levels of any of those things or anything weird we don't expect to find in there. So white blood cells, that's going to be the cloudy part from your UTI.
right? Abnormal colors, blood or blood proteins, those things are going to be pathologic, or at least indicate a pathology for us, right? So we use urine quite a bit as a diagnostic tool. We don't use it like we used to use it, like in the Middle Ages, but we still use it quite a bit. So you're oftentimes going to be asked to pee in a cup, which is part of the deal. Okay, so that then kind of puts a, I'm going to close off your kidney function, and we're going to get to the remainder of the stuff, just the plumbing, which is not particularly exciting, but it is what it is. So right after we leave the renal pelvis, we've exited the kidney at the hilum, we are now in the ureter. And the ureter, like every other hollow organ in your body, has at least 3 layers or tunics to them. We have a mucosa, right, that inner layer. Remember that your urinary tract is exposed to the outside world. So the membrane on the inside of it is a mucous membrane, so we have a mucosa. We have a muscular layer in the middle, the muscularis. And then our outer fibrous layer here, because we are retroperitoneal, it is the adventitia. From the ureter, then we drain down into the urinary bladder. Here we have temporary storage of urine. Again, this is retroperitoneal. This entire process is retroperitoneal. There are slight differences here between males and females. Males have a prostate gland, which sits just below the urinary bladder, and we'll see that in the next chapter here. Females, biologically female people, have no prostate gland, and so the urinary bladder simply sits essentially on the pelvic floor. The bladder itself is not particularly interesting. It gets very, very flat. It's thin-walled. So if you do a cadaver dissection, for example, oftentimes, particularly in females, but also in males, it's extremely hard to find because it is so thin, so flat, and basically just plastered to the pubic symphysis. So it's not a very big thing in any way, shape, or form. But when it does get fully inflated is the wrong word, but full, it can get up to about 12 centimeters long. Normally in class, this is where I'd hold up my hands and show you, but obviously I can't do that now. 12 centimeters is pretty impressive, and it occupies quite a bit of space in your pelvic cavity, which is, if you've ever had a full bladder, and I assume you've all had a full bladder at some point, part of that discomfort, right, is because it's big. It's holding half a liter, and there's a half a liter of space now occupied in your pelvic cavity that didn't used to be there. Now, in an emergency situation, we can actually double this. We can hold up to roughly about a full liter. Now, we don't want to do that over the long term, right? That actually is pretty dangerous. So you're in your car on your road trip, and you've got your giant big gulp, and you drain the whole thing 5 minutes from leaving 7-Eleven. You really have to pee at this point, right? The driver of the car needs to stop and let you over, because particularly with the lap belt situated where it is, a sudden stop can actually put enough pressure on that extremely full bladder to cause it to burst and to rupture, and you absolutely do not want that to happen. As long as urine remains in here, it is not necessarily sterile, but again, not terrible. But you let this out into your pelvic cavity, and all of a sudden you've got an infection brewing, and that is bad, bad news. So a burst bladder is not great. Here, just in this little cartoon here, so here is the bladder itself, the trigon of the bladder, this little funnel part at the bottom of the trigon sits there. This bladder is empty, so you can see these little folds, rugae, inside. These are just going to be there to allow it to expand without ripping. Because we have a male cartoon here, we have this guy right there. This is the prostate gland just below that. And this trigon dumps into the final passageway out here, which passes, in this case, through the prostate, which is the urethra. Okay. Again, this is not particularly exciting. It really isn't. Really, the only exciting part about this here are the sphincters. Just like with the digestive system, where we had an internal and an external anal sphincter, the internal one being involuntary, the external one being voluntary. allowing you to choose when you defecate. Here, these internal, external urethral sphincters allow you to choose when it's time to micturate, which is the technical term for this. The urethra itself is slightly different depending on sex. The female urethra is very, very short, right? So here is the pelvic floor, right? The muscles of the pelvic floor. The bladder sitting right on top of that. So we have this relatively short, about 3 to 4 centimeter long urethra, okay? Not much. The male urethra, on the other hand, is a little bit longer. There are a lot of different things that go on with it. We have to pass through the prostate, number one. So we have a prostatic urethra, which is not very long. It's about the length of the female urethra in total, okay, about 2.5 centimeters, a little bit shorter. And then we have another 2 centimeters that we call the intermediate part, where we connect the prostatic urethra to, essentially, to the base of the penis. And then about 15 centimeters there within that penis, that is the spongy urethra, and that's just, go back here, that's this part. All right, so the spongy urethra, intermediate urethra, prostatic urethra. Because we have to pass through all those additional little parts, that's why it's a little bit longer. Okay, all right, last thing here. Micturition itself, this is the fancy term. If you've seen The Big Lebowski, you've seen this term used before. The more common term here is urination. On a chart, voiding, okay, whatever. Again, this is just the process of getting urine out of your body. 3 things have to happen here. We have a muscle here called the diffuser muscle, right, which I'm not super worried about you remembering. And then beyond that, we have the internal and external urethral sphincters. The internal, remember, this is autonomic. You don't have a choice about this. So when you feel like you really have to pee, this is the relaxation of the internal urethral sphincter, which puts pressure on the external urethral sphincter. When you actually urinate, when you actually micturate, when you actually void, whatever, that's the opening of that external one under conscious control. Okay, so that's the urinary system. We'll close it off here. And then in the next and final set of slides, we'll talk about the reproductive system and a little bit about development.